Okay. We will actually start at the beginning here. I think some of you guys we didn't get this far yesterday. So let's talk about impulse transmission and action potential. You guys can turn in your books to page 203. And again, I really want you guys to read the little section on action potentials right here. So make sure that you guys do that. I want you to do that while it's still fresh on your mind. Okay, so impulse transmission. Okay, so we talked about the action potential itself, how the impulse actually moves in the neuron. But let's talk about how it can move between neurons and how it actually moves between neurons and then even at the axon itself. Okay, so there's a couple things that affect how fast an action potential can move. Okay, there's actually three, but two main ones. One is a myelin sheath. Okay, and if you'll remember, if we go back, actually, you guys will turn to page 200. There's a picture at the top of your book there. And I'll go back on here. We'll look at this one. This is a different picture than the one that you're looking at, but I want you to look at the one on page 200. Okay, if you'll notice, we talked about Schwann cells the other day, and hold on a second. Let me close out some things. I think it's making it run a little bit slower than it needs to. Give me just a second. Okay. I think we're okay now. Okay, I'm sorry. It's just said it was moving kind of slow because I had too many things open. So we'll fix that. So anyways, I want you guys to look at this picture on page 200. And Schwann cells is something we talked about the other day. These are those cells that kind of look like, I think I said Swiss rolls. And so Schwann cells they're just cells that actually insulate an axon. So if you'll look at this, this is a Schwann cell, okay? This is actually a unipolar neuron, okay? And if you look at this, if this is the axon over here, okay, these are Schwann cells. And on here, it kind of looks like they just like cup around the axon, but they actually roll around the axon, kind of like you would roll up, a cinnamon roll or something. So they're actually rolled up and they insulate these axons, okay? Well, collectively, these Schwann cells form what we call the myelin sheath, okay? They're myelin. What that means is, or you'll hear the word myelinated, that means this neuron is myelinated because it has a myelin sheath. Okay, well, when we have these Schwann cells and they are on this axon, they also, the little spaces in between them, which are just the axon, okay, is just where you can see it in between these Schwann cells. Those are called the nodes of Ranvier, okay? So these are just nodes of the axon. So what happens is when an action potential actually goes through a neuron like this, the action potential actually jumps across each of these Schwann cells. So like and we're talking about the whole potential where it gets polarized, where the action potential, depolarization occurs. It occurs only in these areas where the nodes are, and it just jumps across. Okay, kind of like same idea as electricity, like it would jump across water, if you want to think of it that way. It's like because it's an insulator. Same idea. So that nervous impulse jumps from here to here to here to here. Okay, that the conduction of an impulse by way of jumping across these Schwann cells, that's what saltatory conduction is. Okay, so when you read this where it says saltatory conduction, that's what it's talking about. An action potential that actually jumps across those Schwann cells onto those nodes. Okay, that's what it's referring to. Now that is the fastest way for an action potential to be transmitted. Okay. Now the other thing that factors in on impulse transmission is the diameter of the axon. So if you have an unmyelinated axon, so an unmyelinated neuron, one that doesn't have those little cells on it, those the myelin sheath or the Schwann cells, okay, one that's unmyelinated will move, the action potential will move a lot slower in those, but it will move faster with the larger the diameter. So the larger diameter the axon is, the faster the action potential will move. Now, it's still not going to be as fast as a myelinated one, but you get the idea. Okay. One other thing that can influence speed of an impulse transmission is body temperature. Typically, the warmer your body temperature, the warmer those ions can move across that threshold. One of the things that it says in the book is that as far as nerve impulses and how fast they can travel, how fast they travel is, says impulses that signal limb position to the brain travel very fast, up to 119 meters per second. Okay, whereas a pain receptor, it moves a lot slower, less than 1 meter per second. So that kind of explains why, like if you stub your toe or something happens, sometimes it takes just an instant for you to feel it. It's like you don't feel it at first, and then all of a sudden, bang, it hits you. Okay, that's the idea behind that, is that pain travels a lot slower than like me being able to move my arms. Okay. Okay, transmission at the synapses. So we're talking about transmission between a neuron and something else. So it could be, as in this picture, it could be that exchange of an action potential from a neuron to a muscle, like we talked about in the last chapter, or it could be from one neuron to another neuron. Okay, so there's a few different ways for transmission to occur where neurons meet up. The first one is called a gap junction. Okay, now a gap junction is actually just like it looks like a little gap between two muscle cells, and this is specifically refers to cardiac muscle cells. Okay, that's why they're able to contract, I'm sorry, I can't talk, so rhythmically, because they can move electrical impulses very, very quickly. It doesn't have to go through this whole process. It can jump across that gap junction from cell to cell. That one's kind of special. Okay, now the next one, neurotransmitter, the next term underneath here, I'm talking about this particular area. We talked about a specific neurotransmitter in the muscle system. That was acetylcholine. Okay, so acetylcholine is the neurotransmitter specifically for muscles, but there's other ones that take place between neurons, like serotonin, and we'll talk about some different ones. So these are some ideas. I just want you guys to have an idea, but a neurotransmitter is just a little chemical, okay, you can kind of see one across here, that transmits a message between the neuron, the axon terminal of one neuron, and either another neuron or a muscle. So the synapse, remember, is that gap between those, okay, is the gap between those. And the neurotransmitter is just the little chemicals that travel across that gap into whatever tissue it's trying to connect to. Okay, and there's a couple ways that this can work. One is an excitatory effect. Okay, an excitatory effect, that means that this impulse is going to cause whatever it's going towards, like in this case a muscle, it would cause it to contract. It's going to make it do something. It's going to make it, kind of think, like feedback. It's going to make it do something, like contract in a muscle. Okay, inhibitory effect is going to try to make it stop. So an example of an inhibitory effect of transmission is like when we get endorphins. When we start undergoing pain, our body creates endorphins, and those endorphins try to inhibit pain. That's kind of like that idea. You've probably all heard of, like, you do exercise and it causes you to have endorphins, and endorphins make you happy. Okay, that's that idea. Endorphins help to inhibit pain. So that's an inhibitory effect. So, pretty cool. Okay, I want you to write down 6 steps that an action potential takes to go across a synapse. Okay, so you can write these down on your paper somewhere. 1, an action potential reaches the terminal, and remember, an axon terminal is just the very end of these axons, very tip of those, and the terminal depolarizes. We've already talked about that. We know what depolarization is. So the terminal, it's traveled all the way to the very, very, very tip of the axon. That's the axon terminal, where it ends. So depolarization occurs at the axon terminal. Number 2, calcium gates open and calcium rushes into the terminal. So that depolarization causes, at the very end of this neuron, of this axon terminal, calcium gates open up and calcium rushes in to the axon. Okay, the third thing that happens is that vesicles inside the terminals then they attach to the cell wall. So these vesicles right here start moving down to the end of the axon terminal, and they attach to the wall. And this is called exocytosis. We talked about this at the very beginning of the year. We talked about different ways that things move in and out of the cell. This is by way of, this is a type of, it uses ATP, so in its process. But anyway, so it attaches to the outside of the cell. And then number 4, neurotransmitters diffuse across the synapse. So number 4, neurotransmitters diffuse across the synapse. Okay, so let me go back over the first 4 steps, and then we'll get to the last 2. 1, action potential reaches the terminal, and the terminal depolarizes. Number 2, calcium gates open, and calcium rushes into the terminal. Number 3, vesicles inside the terminal attach to the cell membrane. Number 4, the neurotransmitter is diffused across the synapse. Number 5, the neurotransmitters move to the neuron or the muscle fiber, whichever one they're going to. Neurotransmitters go to adjacent neuron or muscle fiber. And number 6, enzymes remove the neurotransmitters. Okay, so you think about it. Think about that last one. So we have neurotransmitters, if you look at this picture, that float across here to this muscle to tell it to contract. Okay, well, the problem is some of those neurotransmitters are not going to make it. May not even need all of them. So you've got all these extra neurotransmitters in here. And if you think about it, that would be not so good, because then as soon as this muscle quits contracting, if you have all these leftover neurotransmitters, they're going to get plugged back in, and it's just going to keep contracting. We don't want that. Okay? We don't want that at all. So we have enzymes that actually come in and clean this up and get those extra neurotransmitters. Okay? Pretty interesting, I think, anyway. Okay, so let's go on to reflexes. I would also encourage you to read this little section where it talks about transmission at synapses. I mean, I would encourage you to read the whole thing. But again, the more you, like, now that you've heard this explained, the more that you read it, the more you're going to understand it, okay? And I would encourage you guys, if it's something that you're struggling with, you're like, I don't really get what she's saying, watch this video again. It's on YouTube, okay? So watch it again if you need to. The other thing is go back and read and take notes when you read. This is the way that I prepare for my lecture with you guys. I take this PowerPoint that's kind of just hits the high points, and I go back in and I read the chapter and I just make notes on it. And that's what I present to you guys. So it's going to be the same thing. This is where I'm getting my information. Okay, reflexes, somatic and autonomic. Okay, somatic stimulates skeletal muscles and autonomic stimulates involuntary muscles. Makes sense, okay? Reflex, the definition of a reflex is a simple, rapid, involuntary, programmed response to a stimulus. Simple, that means it's not complex, okay? It doesn't take a whole lot of pathways to figure out what's going to happen. Rapid, it is very, very fast. Part of the reason it's so fast is because it actually only goes to your spinal cord. It doesn't go all the way up to your brain and go back down. It goes to your spinal cord and whips back around and comes back down into the motor neurons. Programmed response. That means it's supposed to do this. There's one specific way. If this happens, if it's a reflex, there's a specific stimulus, okay? Like, for example, that stretching, like when you hit your knee, for example, you hit it, that stretches your quad. That stretching is sensed with your sensory neurons, goes up to your spinal cord, attached to the motor neuron, comes right back down, and makes your muscle contract. So it's programmed. It's going to do the same thing. It's not like if you hit my knee, my elbow is going to jerk up. Okay, that doesn't happen. So you hit my knee and my arm goes up. That's not going to happen because this is programmed. It has a programmed response. It's supposed to do the same thing. Okay. Well, that's the rest of my notes now. Okay, I guess that's it. But I do want you guys, look at the picture on page 204. Look at that picture of this reflex, okay? So sensory receptors at the finger, page 204, goes up the sensory neuron, that one that picked up the message, goes to the spinal cord. There's an interneuron, there's like an in-between neuron, but it's only in the spinal cord, okay? And then the motor neuron goes back down to the muscles to tell it to jerk that arm back up. Pretty interesting. Okay, and I think that's it. You guys, let's look at these real quick. One says a blank is an all or none response. Okay, and the answer here is action potential. It's an all or none. It's gonna, if it starts, it's gonna keep going. Okay, you can't just make it stop in the middle of the neuron. Okay, number two, saltatory conduction only occurs in myelinated axons. Remember, those are the ones with the Schwann cells that makes it jump from node to node. Number three, reflexes are rapid and involuntary responses. And then number four, the axon terminal has tiny vesicles filled with neurotransmitters. Okay, that's the chemical message that tells it to respond. Okay, so good deal. You guys can do the reflex lab, get that finished up, and then that's it. I'll see you guys Thursday.